a new protein or transcript sequence. In this case, the entire sequence is being shown from left to right, and you can see on that top ruler that we have 170 amino acids total in this sequence. So when we look at something like that little orange blob that appears all the way to the left under predictions, we see that that is a signal peptide. In fact, there are a variety of signal peptides that are all found in the N-terminus of this protein sequence. So we were talking about 5' and 3' as ways to tell the orientation of the transcript. Here we're looking at N-terminal and C-terminal because this is now translated. We're looking at the protein sequence. So all of those little blobs that we see down at the left-hand side represent different parts of a signal peptide that helps this protein know where it's supposed to go after it's translated. I think of this as kind of like the shipping label. In this case, the protein sequence contains within it a destination location. So the transcript is, the translation is produced at the ribosome. The ribosome does lets go of that protein, and then it goes through folding. It may go through the Golgi to be shipped and packed to packed and then shipped off to another part of the cell. And then these signal peptides are essentially the address label, and they get chopped off before the protein becomes functional. So we get all that information here. EnterPro is also giving us some hits to signatures about the family and superfamily of this sequence. Here we can see that the superfamily was 4 helical cytokine, and the family matches say, well, we think this is an interferon gamma. But I would note that we also have hits to the Gene3D database, which means that through sequence, EnterPro was able to say, I know about another sequence that looks like this that is associated with this 3D structure. So that's all grand. We're getting a lot of information back from this. So Gene3D is not just a sequence database, as I've mentioned. It also contains links to 3D structures. These are part of the protein data bank. Here we're looking at a whole bunch of sequences that are very much like this one, that have all been aligned against each other. But we're also given a typical fold into which that sequence falls. So it's possible to do things like say, this sequence is like the others in all but this one amino acid. So that one amino acid may cause a shift in the structure, and so you'd be able to visualize that through this technique. Okay, but I think everyone knows that if you're looking for homology, the elephant in the room is BLAST. BLAST is a hugely popular toolkit, and there are a lot of people who feel that if you're not teaching how sequence alignment and BLAST work, you're not really teaching bioinformatics at all. So it's worth talking a little bit about it. In this case, I'm using a web server for NCBI BLAST that is on the European Bioinformatics Institute, the EBI. They have a lot of really good tools implemented in web form for use. And because EBI is so strongly associated with Uniprot, you can often find a good search against Uniprot is achievable through EBI tools. In this case, I've given this is a BLAST-X run. So the sequence database we're searching against is a bunch of protein sequences. But the sequence that we're feeding to BLAST is a messenger RNA instead. So the software is able to do this 6-frame translation in silico and understand which hits are coming against which reading frames. Now, I realize that the table that I'm showing here in those rainbow colors is really, really small. But I want to draw attention to just the very first interferon gamma sequence that we hit in the database. Does everyone see, it's, as I said, very small, underscore FELCA at the top? At the very top sequence in that alignment. I wanted to point out that a lot of people think that hyenas are dogs. But when we do a search against molecular databases like this one, what we find are a bunch of hits to cat sequences. Felis catus is the house cat. So the house cat's interferon gamma sequence is the closest match to the interferon gamma from hyena. We see a whole bunch of, do I have a chat session? Okay, I just saw the message bar pop up at the top. Okay, so each of these hits is being evaluated by an expectation value. That's this column that appears in between those two rainbow stripes. So that set of E values gives us a way to evaluate, is this match likely to have happened by random chance? Certainly, if you see an E value around 1, that is that's a very dubious match. I got a blinking thing. There are 2 questions showing up in chat. Let me see if I can get that to show up again. It's not actually showing me the chat session because I'm sharing the thing on screen. So I'm going to pause for just a moment. Oh, thank you. It's just people saying they like what they're seeing. I'm glad for that.
The presentation is very nice.
Okay. If I start moving too quickly, by all means, wave your hands frantically at me because I'm not seeing the chat stuff while I'm sharing the slides.
Fine.
Okay. Thanks. Okay. Great. So what we see when we search hyena sequences against the database are not dog sequences at all. Instead, we get hits on the house cat, on cheetahs, on lynxes, on tigers. So the taxonomic people have weighed in on this and said, we feel sure that hyena is more related to cats than to dogs, and the molecular information reaffirms that. All right, now there are a couple reasons why you might find homology between a novel sequence and another. Those usually fall down to paralogy and orthology. Paralogy means that over time, a gene has, over the course of evolution, a gene has duplicated itself so that this species now contains 2 descendants from the same gene that differ considerably in function quite normally. It is also possible that we find relationships between a protein in a human being, for example, and a protein in a blade of grass. We are, after all, related if you throw in enough billion years. So when we are annotating a new organism, we can benefit highly by the huge number of species that we've already annotated quite a lot. So one of the programs that I've come to use quite a lot is this protein ortho software. So if I have a list of putative protein sequences for the hyena, and I want to determine which of them are like other species, it's nice to find homology in general, but it's even better, I think, if I can find the closest relative in another species. So I might say, because we know hyenas are very closely related to house cats, that we're going to use a sequence database of house cats, of tigers, of lions, of lynxes, of panthers, of cougars, etc. And then we can ask which proteins are shared among these species. Specifically, what is the individual gene in house cat that is most similar to this individual gene in hyena, or to this individual gene in panthers? So protein ortho was originally designed around the concept of reciprocal best hits in BLAST, which is to say, if gene A is similar to gene B in BLAST, gene B is also most similar to gene A in BLAST. So it tries to decompose all these relationships among an ortholog, which is to say the same gene in different species, to find the best matching protein across these species that helps us to understand the conservation of this gene across a whole family of related species. So it's very nice. Protein ortho handles a lot of the work of untangling all these relationships that form to try to find individual one-to-one matches, or one-to-many matches even, across species. One of the reasons why it's able to do this quickly is that it doesn't use BLAST, which is, although it is one of the most widely cited tools in bioinformatics, is not in fact the fastest out there now. Computer scientists treat fastest algorithm as a very temporary title. And you can always imagine that they're trying to create some new tool that's going to be even faster than the fast tool of last generation. So given that BLAST was published a few decades back, people have been putting a lot of effort into this. And the diamond algorithm used by Protein Ortho is astonishingly fast. I'm always stunned by how much development takes place in these areas. Okay, so that starts with our first question then. Is sequence homology able to inform us about other proteins like the one we've collected so that we can understand the new gene based on what information we have about the old one that was already in the database? We often want to find information about co-expression. So if you imagine that we have microarray experiments where we've measured the same set of genes over a very broad set of conditions, maybe let's say we're looking at yeast, right? So a lot of people are doing baking given that they're locked down in their homes right now. So imagine that you have a microarray for all of the genes of yeast, and you are measuring them when you grow yeast on lactose, when you grow it on dextrose, when you feed it a little less dextrose than you might have in another case, when you've introduced another microbe into the mix, all these different conditions that you're pulling together. So each of these microarrays that we see here represent a different condition. If you have 30 or 50 or let's say hundreds of different microarrays, each representing a different kind of growth condition, you would expect that genes that show up in the same pathways are likely to be correlated in their expression. That if you dump a bunch of glucose on your cells, the genes that are used to break down the glucose to 3-carbon sugars, etc., will be up in expression to accommodate this new stimulus. So over the space of lots and lots of microarrays, an individual gene is going to be up under some conditions and down under others. And so the colors here represent that. So genes versus microarrays is in this case a case of genes shown across different stimulations. There are a variety of ways that we go after decomposing sets of genes to find those that are most similar in their relationships. So if you saw another gene that had a low in expression under these two conditions and a sort of low over here and a high in this one, you could imagine sorting those together to say, these genes have very closely linked expression values. So techniques like ICA, shown in this paper, this is a paper that hasn't even gone through peer review, but I loved it because it had a visualization that showed exactly what I wanted. So I hope you don't mind that I used one that's just coming out of preprint servers. So in this case, they used ICA decomposition. So one matrix becomes 2 over here. So how does that work? The idea is that genes can be sorted into modules, which is to say genes that have similar patterns of expression over the microarrays get called modules instead. Then we can say, how do these modules compare in which conditions they are strongly expressed in and those that they are weakest in? So in effect, what we're doing is grouping together genes by the fact that they show up under the same circumstances. And by knowing which circumstances turn them on or turn them off, we are getting an assertion about their likely functions. Now, there are lots of ways to go about doing this. I showed ICA in the previous slide. Here we're using regression or mutual information, simple correlation coefficients will get it done, or Bayesian techniques. We also see that people have used machine learning in this case. Machine learning has creeped into basically every area of bioinformatics over the last few years. So it's quite rare that anyone works in a space of bioinformatics that doesn't have some sort of link to machine learning at this point. What we see, though, is that if 2 different tools both use regression as a way to discern which genes are related in function, those tools, even if the software around the regression function is quite different, tend to perform very similarly when we look across different techniques that can be used for the same thing. Bayesian networks tended to fall together on the map, Those that used mutual information and correlation also tended to be close together. So even though you can have very different methods for analyzing these gene expression networks to build out modules based on correlation or whatever, we find that they tend to produce complementary results. And having run a regression-based tool on the data set and then run a mutual information-based tool on the data set, combining those together is likely to give you better data as a result. So in this case, if we look at the performance of an individual engine for analyzing a microarray, we see that the performance is relatively low. Once you add together 5 methods or 10 methods or 15 methods, the amount of overall quality that you're getting out of the resulting gene networks gets better and better. So this is a field where individual engines, none of the individual engines can really claim to best performance. But once you start putting them together, the consensus gives you much better result than what an individual engine would. I think a lot of people see diagrams like these and they say to themselves, I'm not really sure how to interpret that. So I wanted to give you some clues on how to read the biological networks that come out of gene correlation experiments. I'm not sure if students will have had much exposure to biological networks, but when we talk about them, we often refer to them as hierarchical and modular, scale-free, and small world. Which is to say that a scale-free network, like the network that we see among genes, is one that's got a whole bunch of hubs. Individual proteins or individual genes link to very large numbers of genes in the case of hubs. Other genes link to very few others. So what we have is essentially, imagine that you're taking a gun and shooting some gene out of a cell, just saying, I'm now completely abolishing the function of this gene, pow. Generally speaking, you can shoot at almost at 50% of the genes in the human genome and still get a viable cell. But if you're shooting out a gene that's a hub, you're going to have a very different experience. That's because some genes, like, well, these are transcription factors over here, but some of the genes that we see here, like say this blob here, is connected to a whole star of other genes in its local neighborhood. And those hubs are points of special sensitivity for cells. Okay, but the topic that I'm giving here says that this is hierarchical and modular, which is to say that if you look at a small group of genes, you're going to see a modular relationship. These all relate to each other in a very powerful and closely linked way. But generally speaking, as you move it to a higher and higher level, you see that, for example, all of these purple genes associated with tRNA processing are all grouped together. But when we look at just a subset of them, we see that they're more closely related inside the relationships of the bigger cluster. And this bigger cluster itself, the purple region itself, is also linked to translation and to the tryptophan metabolism proteins and so on. So whichever scale you're looking at genes, you're going to find groups of close associations, and then those groups of association are themselves connected to others. Okay. So given that we can do so much measurement with microarray expression and with RNA-seq today, you might think, goodness, well, if everything that we want to know about gene expression is all bound up in messenger RNA. The reality, though, is that protein quantitation gives us very different results, quite typically. So just to give one paper that made a big impact on microarray, this 2003 paper from trying to remember the name of the PI, but Gami et al. From this 2003 paper did an experiment where they were quantifying each protein via Western blot in yeast, and they were also doing measurement of the number of copies of messenger RNA that were visible for it. So what they discovered was that a rank correlation of the protein levels versus the transcript levels only gave an R factor of 0.57, which is not a huge degree of correlation. If you look at this plot at the right, you're seeing their estimated number of protein molecules and their estimated number of RNA molecules. And you don't see a really strong diagonal here. It's, yes, the more copies of RNA, the more protein you have as a general rule, but there are a lot of exceptions to that. So certainly we get information about protein levels that can be quite divergent with what we see in in gene expression levels. All right, so we've now talked about our ability to find related proteins, homologous proteins that are similar in function by BLAST or diamond for that matter. We've talked about gene correlation, or expression correlation, but there are a lot of other methods that we try to use to make assertions about two genes having similar function. One of these routes is co-localization. If you can show that two gene products are showing up in the same part of the cell, you may be able to assert that they take on similar functions. But there are three definitions under co-localizations I want to cover that put some limitations on this. So we start with co-occurrence. If we are looking at fluorescence microscopy data like we are in these the ABC panels down at the bottom of the slide, we're looking in this case at different endosomes within actually a dog cell line, MDCK cells. So if we were to find that everywhere the red dye was present, the green dye was present, we would be able to say that the protein products to which they were linked were showing up in the same locations. You can do that with confocal microscopy. In this case, most of panel A shows yellow colors. So when we're looking at fluorescent dyes, a red light plus a green light looks yellow. So all the instances of yellow that you see over there are places where both the protein labeled red and the protein labeled green are co-occurring. You see in that first little inset box on panel A, though, that we have a distinctly greenish shade, that this region of endosomes do contain the gene that's labeled in C, but they don't contain the gene product labeled in B. And the ones over here are somewhat reddish. So these reflect co-occurrence. These reflect a biased occurrence, that one of those dyes is more likely than the other. So to say that things co-occur is not to say that when red goes up, green goes up. So look at the diagram over in panel D. That's an example of correlation, which is to say that we have a scaling effect, not just that red dyes and green dyes are found at the same place, but where you find more green dye, you also find more red dye. In this case, telling you about the protein to which those are connected. Now, to say that things co-occur, or to say that they correlate, is not the same thing as saying that gene A interacts with gene B directly. For that, you really need to have much more tightly linked information about their co-occurrence. You need something. You need to meet a higher criterion than to say that they're correlated. So there's a fluorescence-based technique called FRET that can allow us to put two dyes together. Well, one dye on gene red and one dye on gene green, and only when those products come together within a few microns do you get fluorescence. That's the FRET technique. You might also use electron microscopy to establish that. But generally speaking, if you're using standalone dyes on gene products, you can establish correlation, but getting to interaction is a lot harder to claim. Bioinformatics has certainly made its way into our analysis of imaging information as well. So in this case, we have some regions that represent cells and some regions that represent background. In this case, we're specifying regions of interest and segmenting these images. So if you were to look at G by itself, it's pretty clear that stuff that are cells, these green areas, stand out from the black background in which they sit. So segmentation is just saying what's foreground and what's background. But then region of interest analysis is subdividing these cellular images into individual cells, because we see that individual cells actually have pretty different levels of expression for the two dyes. So at the lower left in panel E here, we see that this cell is expressing a lot more of the green dye than of the red. And we see some mix of the two showing up in these two cells. So that's really valuable for us to be able to evaluate them. So regions of interest and segmentation are ways of focusing our characterization of imaging data on just those limited areas. Okay, so that's great. If you're using confocal microscopy, you can learn a lot about your cells. I'm going to take just a moment to drink some tea. I can talk a long time, but a little tea helps a lot. Okay, so let's talk about another way that we can go after protein-protein interactions. We talked about FRET, and we talked about electron microscopy. But we would also think about the fact that these protein molecular machines are frequently not just individual polypeptides folded up. They frequently are forming things like homodimers, where two copies of the same protein come together, or heterodimers, where protein A connects up with a copy of protein B. And these relationships are necessary to function. So defining protein-protein interactions is one of these things that we really need to go after. So I thought that this was a really good definition from Las Rivas, de Las Rivas and Fontanillo. So the physical contact considered in protein-protein interactions should be specific, not just all proteins that bump into each other by chance. Cells are crowded places, you know? It also should exclude interactions that a protein experiences when it is being made, folded, quality checked, or degraded. Okay, so if you ask what protein touches others most commonly, you would find that ribosomes touch every protein because all proteins are manufactured at ribosomes. So we do not claim that ribosomes form PPIs with every protein in the cell, because that's just part of making one. Similarly, a chaperonin that is useful for helping to guide a protein to its final conformation is not considered a protein-protein interaction by this. And if you have a protein that is targeted for ubiquitination, for degradation in the proteasome, we don't claim that the ubiquitin, the E3 ligase that sticks the ubiquitin onto the proteins is forming a PPI. So generally, we're talking about the functional life of the protein, not its birth or its death in that way. Okay. Is it possible to clear away? There's some writing on screen. I'm not sure how to erase the red mark there. It's okay. We'll just move on. So the yeast to hybrid system is one of the most widely used methods from say the late 1990s and 2000s. It was an approach where we could clone new genes into yeast and ask, do these gene X and gene Y interact specifically in the context, do they form an interaction within these yeast cells? So here we see that we are creating a hybrid fusion. We have the X gene being linked with the DNA binding domain, here marked BD, and we have an AD, that is a transcription activating domain that's getting associated with gene Y. So if gene X and gene Y form a heterodimer in the cell, if they come to form a strong interaction between them, that also means that the BD and the AD are brought together. If that happens, then you get a reporter. And that is one way that we can establish that gene X and gene Y interact. Okay, so that's very commonly used. It's got some problems, though, with forming a lot of false positives. So it's also valuable that we can do our assessment not on a gene level, but on a protein level via co-immunoprecipitation. I got this slide from my friend Alexei at University of Michigan, so I'll try to walk through this. We think that if we were to pull a protein B, here B stands for bait, if we pull the bait out of a cell, we would imagine that proteins with bait protein interacts will come along for the ride. Okay, so if I pull on B, all those P proteins, the prey proteins, come along as well. So generally speaking, if I can introduce a tag into my bait protein, here marked T, and grab it out with some sort of antibody on a solid support, I'm able to grab along all those prey proteins as well. In reality, we find that contaminant proteins like sticking to our solid supports, contaminant proteins like sticking to our antibodies, contaminant proteins like sticking to our tags, they like sticking to our bait proteins, and sometimes even the prey proteins can come along with their own contaminants. So that obviously is going to complicate making sense of these data. But co-immunoprecipitation is a very widely used method for understanding cell biology. If you have a particular protein and you're trying to understand what its colleagues are in that interaction, co-IP is definitely one of the most popular routes. Now, it's also possible to create two different kinds of tags used to pull out a particular set of cells. So if I'm using just streptavidin to pull out a biotin tag, that's nice, but it might be valuable if I can do 2 rounds of purification. That I've got a pair of tags attached to the protein. I use one to grab the whole thing out. I cut that away and then capture out the things that have the second tag. Now I have a cleaner set. There are many, many ways to go about doing tandem affinity purification, though. So this article examined which of the methods are most popular. So we see that his tags are very useful if you've got an IMAC columns, because now a protein that you've stuck a whole bunch of histidines on can be isolated out of a mixture, grabbing out that particular protein. Stop the share for just a moment. Hi, everyone. Nice to see you again. And I'm going to put that back up because I think that's going to erase the marks on screen. Good. Okay, good news. So let's move ahead to the next slide. Okay. So we're learning a lot about protein structures along the way. We have a very large number now of protein structures that we've solved for our databases. Is it possible, if you know the structure for a protein, to be able to say that it obviously interacts, it forms a dimer with this other protein based on structural information alone? It seems like that should be feasible, but protein structure has been an area that has lingered behind what's possible quite a long time. There are a lot of things that make protein structure analysis very, very difficult. So structural data, an evaluation of this method was published back in 2010 that helped to reveal some of the problems in it. Localized regions on protein surfaces are conserved among structural neighbors that participate in protein-protein interactions. So let's think for a moment about antibodies. An antibody sticks to a protein not because of the entirety of that protein structure, but just because of the epitope on it. The particular region of the protein for which this antibody is specific is a relatively small portion of its overall size, right? So what we see is that these interactions between protein interfaces are also not whole protein in nature. They usually represent some region of anchoring between these two sequences. Okay, as long as structural information is available for a given pair of proteins, the set of template complexes available in the current structural databases can be used to generate coarse-grained models of protein-protein interactions. So if we imagine something like yeast, yeast has 6,200 different genes in there, and a slightly larger number of protein products that relate to that. Do we have a structure for every protein in yeast? Not even close. Not even close. So starting with the fact that you have to have a structure available in order to determine whether these interactions is available, is likely for a pair of genes, is not frequently a requirement that we can meet. But it is becoming possible, if you have large numbers of structures to work from, to begin inferring these patterns of interaction. Okay. A lot of people, I think, have come into understanding that biological pathway and biological network information is becoming widely available for a very large number of species. It is also the case, though, that which species are covered in a particular database can be quite different. I think many people have heard of GO categories, or GO SLIM, something like that. That is the gene ontology biological pathway collection. Gene ontology has done a really good job of including lots and lots of different species. So if we look at these 6 species at the bottom, we're looking across a fairly wide amount of developmental space. So Arabidopsis thaliana is a plant. C. elegans is a worm. Drosophila melanogaster is a fruit fly. Homo sapiens is us, of course. Mice are musculus, and Saccharomyces cerevisiae is a yeast. So this is a fairly broad sampling of different model organisms that we use. If you look at gene ontology, you can see that the 2 species with the most information within GO are human and mouse. But that is not to say that gene ontology has really neglected these other species. Something like S. cerevisiae looks very small in gene ontology. You see this tiny little green bar down here? It's not a huge fraction of gene ontology. But we should also keep in mind that S. cerevisiae has only 6,200 genes. So the number of interactions within it is much smaller overall than human, which has 20,000 genes, like more than 3 times as many. Okay. A lot of people, I think, also use KEGG pathways, the Kyoto Encyclopedia of Genes and Genomes. What we see, however, is that certain species, well, I would say that their coverage across species is actually quite good. They haven't totally neglected one of these 6 species in favor of the others. But look at what we see with MINT and with IMEX and with wiki pathways. What bar dominates those 3? Human. These 3 databases are brilliant if you're working in clinical pathways or clinical biological networks. But if you are trying to look at Arabidopsis thaliana with those databases, you're not going to find very much in any of those 3. Once again, we see String, another database that a lot of people use for pathways and networks, has a pretty good assortment. It's got an even smaller fraction of all of its database records representing S. cerevisiae, but it's not totally neglecting that. Okay, so what I would point out that these 6 databases that I've highlighted are hardly the only ones you could be using for biological pathways and networks, though. This is one of these are just 6 of the resources that I could find at pathguide.org. If you are working in a particular species and want to know which of these pathways feature information about it, I suggest you go to pathguide.org and just enter it. You can find which of these databases serve that species well. Okay, lots of takeaway messages, and I realize that spending only 50 minutes on all of those is a little unfair, but I want to leave lots of room for questions, and I think we have the room for 35 more minutes. So, so learning a transcript sequence is great. It allows you to leverage considerable sequence resources, and our ability to find homologs is one of those areas where bioinformatics has hit it out of the park. We have great ability to find sequence homology across very long evolutionary differences. We can discover the relationship of human genes to genes in blades of grass, and that is not a close evolutionary relationship. Expression data have been very, very useful for us to understand co-expression partners. This is quite valuable because if you find that gene X, Y, and Z that you know very little about grouped together in their expression patterns with another gene that is well characterized, you can begin to infer that the pathway in which the well characterized gene plays a role may be the same for these genes X, Y, and Z that correlate with it. Co-localization is a hugely valuable method, and it's not our only route to getting to protein-protein interaction data. We require experiments to fuel that. That's not a bioinformatics only route, but large amounts of data have been made available in proteomics to support this kind of assessment. Pathway and network data are always kind of lagging behind, right? It takes a while for functional information to make it into these databases. But we find that at least for human and for mouse, there's very broad support for their biology in these databases. And it is possible to find a resource for understanding pathway and networks in most species that have significant amounts of molecular data in hand. Okay, I'm stopping that share. We are open for some questions, so I would be glad to see those questions. Should we use the chat bar for entering questions? Is that the best way?
Participant have any questions, please type in chat box. Have any questions?
I would like to open this up a little bit. I appreciate questions asked about the slides, but it is completely okay for you to ask other questions about careers in science, or even stuff like, why did David Tabb move to South Africa? I'm happy to answer those questions too. So I'll watch for those to appear.
If you have any queries, participants, if you have any queries, please ask in chat box.
There's one. Thank you. Can we use these sequences to start synthesizing a DNA to combat diseases? Okay, well, I think a lot of people's minds are on the COVID-19 outbreak. And a lot of people think, well, goodness, if we could just sequence some of this, we would have our answer. I would point out that since February, there have been many, many, many copies of COVID-19 sequenced. So at present, we have not just hundreds, but more than 1,000 distinct sequences that have been observed from COVID-19. The primary way that that's been useful so far is, first off, to nail down the relationship of this virus to previous ones. I think most people throughout Southeast Asia have heard of SARS, right? So the first SARS epidemic is a very closely related virus to this one. But there's another outbreak related to this in the Middle East that's called MERS. So MERS and SARS are very much the most closely related ones. It was because we could sequence the virus behind COVID-19 that we knew exactly what relationship it bore to those existing diseases. Today, we primarily use the sequence of the virus as a way to recognize when a new copy of that virus has been brought into a population. So if you look at patterns of COVID-19 in the United States, you see that there are frequently multiple copies. There are frequently many different strands of the virus that have made it into a location because people have been moving around so much in the United States. However, in a place like South Africa, when we had our first outbreaks here, we were able to discern that it was a strain that came from Italy, not directly from China, that had made its way here. So yes, we are doing a lot of sequencing to help us to understand the COVID-19 virus. Now, the question said, can we start synthesizing a DNA to combat disease? That is a little harder. How do you know which sequence is going to produce a gene product, for example, that is best able to combat a disease. That's a much harder thing to do. You could, for example, create an inhibitory RNA. So if you were able to do silencing, in effect, to say this RNA is going to hold back expression of a necessary viral protein, that might be a valuable thing. But I would point out that having a piece of DNA as a treatment is not a very good option for us. Our ability to dump many copies of a gene into cells as a therapeutic is nearly non-existent. So DNA is our route to understanding disease. DNA is not generally a method that we use for correcting a disease or, excuse me, or blocking someone's ability to get a disease. All right, I'm going to move on to the next question here. Thank you for saying it was too good. I love that. How come we know among the transcripts which one will work for a real gene? Okay. So dealing with RNA-seq in a non-model organism is really hard. It's not for the faint-hearted. First off, if you're working with a non-model organism, you may not have a lot of information on related species. For example, if you're working in insects, the number of completed insect genomes we have is really small. So it may be that your closest homologous species with homologous sequences is very distantly related. So, all right, so when you've produced an RNA-seq experiment from a non-model organism, you typically have to go through a process of assembly in order to build a transcriptome from the reads that you've produced. And that's feasible with software like Trinity. But I would note that the amount of RAM you need to do something like that is extensive. So a reasonable desktop computer in the lab has something like 16 gigs of RAM, but that's nowhere near enough to deal with an RNA-seq data set transcriptome assembly if you're using software like Trinity. So when we do assemblies, we typically use a machine with 96 gigabytes of RAM. And that's just not that common a machine out there. So doing the assembly of transcripts is great. Then you need to do, as you've alluded, you need to do some process of clustering to recognize that many different transcripts with one-off sequence variants may be found within your assembly. So something like CD hits becomes necessary. Then you'll probably want to be able to say which of these things produce reasonable translatable transcripts. So you could ask, for example, are there any reading frames here that exceed 50 amino acids in length? If you have a transcript that doesn't produce even 50 amino acids, it's less likely to be a viable protein coding transcript. And then how do you know which of the transcripts that meet all these criteria that are real? Well, that's largely a process of functional annotation to be able to say, this transcript is very much like this transcript from another species. We therefore say this transcript is real, and we have that it's been conserved through speciation, and that it is known in other species to produce a protein. So that's the start. Is it likely, how do you know that a transcript you've made from an RNA-seq experiment comes from the organism you were sequencing? It's obviously, every organism plays host to others, right? I mean, if you were to sequence a region from my gut, you would find not just human proteins, but a whole bunch of microbes as well that live there as a natural, normal part of my digestive tract. So it's possible that in doing RNA-seq on me, you would actually develop transcripts that come from microbes, not from me. Okay, so lots of problems associated with that. That's a very good question. Oh, thank you, very useful, good. All right, how for dry lab protein-protein interaction exactly reflects in a biological system? Okay. Okay, generally speaking, individual polypeptides corresponding to a particular transcript from a particular gene, individual polypeptides do some things in our cells, but more frequently, we find that different polypeptides come together to form an intact protein. So things like hemoglobin, there's not just one gene for hemoglobin, there are multiple genes for hemoglobin because different chains have to come together to form hemoglobin. So we don't refer to something that comes from a ribosome as a protein in itself generally. The protein is what we refer to as what happens when these polypeptides come together to form the quaternary structure of a protein. All right. So if you're going to understand how necessary protein-protein interactions or polypeptide interactions are in a biological system, I'm trying to think of what would be a really good model to work with for teaching this one. Well, let's think about something like EGFR signaling. So epidermal growth factor is a signal that a cell receives at its surface to trigger cells to begin proliferating. Okay, so EGF arrives, it touches EGF receptor in the cell surface, and that kicks off a cascade inside the cell. Generally speaking, this cascade begins by a phosphorylation event. We have a protein associated with the cell membrane that's kind of just waiting for messages to arrive. When it does, this cell is this protein becomes phosphorylated, and once it is phosphorylated, it forms an interaction with another copy of that same protein that's also phosphorylated. Once they're together, they're able to kick off a cascade of phosphorylating other proteins within the cell that then phosphorylate other proteins that phosphorylate other proteins that phosphorylate other proteins to send a message, now is the time to begin growing. So we very frequently find that the formation and dissolution of these relationships is very essential to all sorts of biological processes, not just that one. So think about the ribosome, for example. Ribosomes have kind of like the big part and the small part, and they come together on the transcript to begin this process. So this type of formation and dissolution of relationships is almost always reflected in any biological function. Okay. All right. How is this information going to be useful to you? It's going to be informational to you in a lot of different ways. I would start by saying that, especially in the pandemic we're facing right now, there are a lot of ideas out there masquerading as science. I think all of us have a relative who uses social media explicitly to share conspiracy theories. As scientists, we are training ourselves how to be skeptical about our own data and skeptical about things we read in the press or things that we read in magazines, certainly things that we read on Facebook. The more you understand about how biological function is attributed to sequences, the better you will be at being able to refute nonsense that people spread about how they know what this gene does. So I feel like that's going to be, that kind of information is going to be useful to you. I hope you don't mind if I do just a quick moment of self-promotion here. I have been recording lectures. Let me see. Just a second. Lots of lectures on bioinformatics here. Okay. Oh, I sent that to Inspiring Millions. I guess it gets forwarded to the list subsequently. Okay, so I've been creating a I've been trying to put as many lectures as I can on how to do bioinformatics, how to do biostatistics, and so on a website that I've created. I just realized that over time I developed this huge backlog of YouTube videos, and they span quite a lot of topics. So when I teach bioinformatics at my school, I teach all these different lectures. So you can see that the slides are available in PDF form, and then you can watch the lecture on YouTube over at the left. So we have lectures on that, on stuff like how we do image analysis. My specialty is proteomics, so this stuff is here. And we even have things like a class on biostatistics that I taught. So lots of information available there. This is over at my blog called Picking Up the Tab. So I hope that we can get that information shared to folks. But it's my belief that as scientists, we need to arm ourselves with lots and lots of information. We need to be able to understand enough of what's going on in other fields connected to ours that we are able to benefit from that information correctly. So I hope that is that answers the question appropriately. Okay. Okay. With the amount of in silico studies we have, will it be possible to predict the nature of the action of microbes in simulation? Whew, well, that's a tough one. Okay, so I spent my postdoc years, started from 2003 to 2005 at Oak Ridge National Laboratory. One of the things that I was doing there was trying to help them interpret huge amounts of protein-protein interaction data in microbes. We were working with something called our palestris, basically. It was a bacterium that lives in soil. So trying to understand how that one bacterium acts by itself, sitting in a petri dish, is hard. Trying to discern how Rhodopseudomonas palestris behaves when other bacteria are in the same space is actually quite a lot harder. So being able to simulate entirely all of the pathways of a bacterium at once in isolation is already beyond what we do very well. There are some people who've created a field called flux analysis for understanding how the different reactions taking place within a cell affect each other, right? So if you have something that requires the product D, and you start with product A, and from that you make product B, and from that you make product C, and then finally you can make product D, trying to understand how shortages or increases in different parts of that molecular network lead to changes in phenotype is a pretty difficult thing to do. So I'm not saying it's never going to be done, but I think that this has been a difficult area to work. So simulating simulating a pathway is interesting and valuable, but it may be it may be beyond what we are currently capable of doing. Simulating all pathways in an individual microbe is a very difficult thing to do. Okay, thank you so much for these nice words about this. Okay, is the ITS region quite enough to identify fungi? Ah, very good question. So we didn't cover the microbiome in this talk. I have a lecture that I teach on amplicon informatics that is likely to be of value for this. It's already on YouTube. The PDF of the slides are already available. I'm going to pull this particular slide open from it. So I'm now looking at a different slideshow. This is not the one that we just looked at. So I'm going to share just that screen. Okay. What we're looking at is kind of a guidepost to how we go about studying microbiomes. often people study 16S rRNAs as a way to understand which bacteria are present in a sample. They do this because the bacterial gene for the 16S rRNA has some regions that are very highly conserved and some regions that are very, very specific to each bacterium separately. So we create primers against different regions of the 16S rRNA as our guide to first PCR out a bunch of 16S rRNA genes from all the different species in the sample, and then we can use that in in tools like Mother, for example, to discern which different species are represented there by the different sequences we see. Fungi don't have the same genes, right? They're not bacteria. They're eukaryotes. So instead, we could target the 18S rRNA for fungi or the 28S rRNA for fungi. But I know that some people have targeted these internal transcribed spacer sequences, which are pretty good for evaluating the fungal diversity in a sample. I think that, I'm trying to remember if it was this Sinha et al. Paper or the Taylor et al. Paper that had the treatment of that. I forget which one it was. But I think that that reading one of those papers would probably be a good idea for understanding what the ITS offers for understanding the diversity of fungi. So either Sinha or Taylor, and then those are the journals in which they appear. So it's not the topic of microbial diversity is not really part of this lecture, but I have previously recorded a lecture on amplicon sequencing. You can just find it under my name on YouTube or at that index website I was sharing just a moment ago. All right. All right. Bayesian networks. Let me see. Okay. Oh, here's the $1 million question. That's a much more than $1 million question. When will COVID-19 come to an end? Okay. Dave Tabb does not have a Nobel Prize, so you should take that into consideration as you consider this answer. The current round of COVID-19 will stop being a problem when everyone can be vaccinated against COVID-19. I think that the claims that COVID-19 will go away as soon as it gets warm neglects the fact that COVID-19 was spreading just fine in the southern hemisphere while it was still cold there. We're falling into winter now. It's been hot. The idea that North America will suddenly have an end to COVID-19 just because summer comes on is very deeply flawed. So we should not count on the weather to save us. We haven't created a vaccine against MERS. We've instead been able to recognize people who are exposed to MERS early enough that we can prevent them from being infectious agents to other people. The SARS-CoV-2 virus that causes COVID-19 is a lot harder to beat than the original SARS because lots of people don't get sick enough, they stop walking around and spreading it to other people. So what we really must have is a vaccine against SARS-CoV-2, against the virus that causes COVID-19. I see that people are dismayed that public events for July are now canceling, saying, well, maybe everything's going to be fine by the end of June. there's no reason to think that everything's going to be fine by the end of June. South Africa was one of the countries that locked down really early, so that people were not allowed to leave their homes except for like 3 or 4 allowable tasks. And as a result, as soon as South Africa locked down, transmission of the virus dropped to very low levels. But we also know that locking down South Africa cannot be a long-term solution because people are starving. There are lots and lots of poor people in South Africa, and they don't have a deep freeze and a refrigerator to store a month of food. That's just not something that they can do. It's not going to be something that changes either. So our only possible solution is to be able to track the spread of the virus, and that means slowing down its transmission enough that we can get ahead of this thing. Even so, it's clear that with people traveling among different countries, that a visiting scientist coming to see us from the United States, for example, could bring a fresh copy of the virus without knowing it and infect people here, and we'd be right back off to the races again. So when people, I feel entirely inadequate to answer the question, when will COVID-19 come to an end? But what I would say is that you should expect that the entirety of 2020 is consumed by the world's fight against this virus. Some places are already at a place where they can function again as a society. If you were to live in New Zealand, you would be having a completely different experience of this virus than if you're in, if you're in China or if you're in the United States. I mean, the United States seems really intent on letting this thing spread like wildfire. And I feel terrible saying that because that's where I'm from. But I think that until we have a vaccine, COVID-19 is going to be something we're battling all the time. Let's consider another scenario, though. We have a vaccine against the flu, but that vaccine against the flu is not proof against every strain of the flu that may be circulating in a given year. And we have to put together a new vaccine every year to reflect the changes in that virus. This might be one of these situations where COVID-19 is not just making an appearance around the world in 2020, but also in 2021 and in 2022, each time with a substantially different sequence. If it's a case like that, we're going to have to get ahead of this thing by having a substantial infrastructure for making new vaccines yearly for this particular virus. That's how COVID-19 comes to an end. It doesn't have hosts to infect anymore. Herd immunity would make subsequent years much less infective than this year. But right now, because nobody's immune system has seen it, it's incredibly infectious. So further years, we would have better infrastructure for dealing with it. But this is a tough one to beat. Sorry, I wish I could give you a much happier answer on that. But we have an awful lot of scientists throwing away their old research programs and moving into COVID-19 research because everyone knows how important this is. Everybody wants to protect grandma and grandpa, and frankly, ourselves. You know, I'm 46 years old. So 46 might seem like, oh, I'm not in a danger zone at all. But the reality is, if I have to go into ICU for treatment, I'm really sick, and I will probably have ongoing effects from year to year. All of us are highly motivated to do all we can to beat this disease. Okay, I'm going to move on to the next question. I feel like talking about COVID-19 gets me really head up, so I'll try to keep calm. Can Bayesian networks correlate gene and environment and disease? Now, I made reference to Bayesian systems for detecting correlations of expression from microarray data. That was part of the second part of today's talk. Being able to do people know about Koch's postulates? There was a famous researcher, Robert Koch, who put together a series of postulates about how you would know the causative agent of a disease. So his postulates are the kinds of rules we use for saying, we observe this disease, we are confident that this is the agent that causes that disease. So generally speaking, his postulates have been very useful for us to understand things like the fact that COVID-19 is linked to SARS-CoV-2. We also see that kind of relationship derived through scientific principles is sometimes ignored. So South Africa, before I lived here, had a president who got bad advice that the HIV does not cause AIDS. So his public policy health direction was that you need to take these traditional medicines to heal you of AIDS. And so a lot of people in South Africa had untreated HIV infections for a very long time, and it was quite dangerous. So just because we have scientific information that is credible about the causative agent of disease does not mean that society will act on it well. So Bayesian networks are kind of from a different side of the biostatistical spectrum for me. I tend to work more in the frequentist domain, which is kind of poor because Bayesian networks are very, very powerful. So I would say that understanding the relationship between gene expression and the stimuli that organism is experiencing is certainly feasible through Bayesian methods. And when you have copies of the slides, you might look at the one that talked about different routes to discerning these relationships. I'm going to re-show that slide. Here we go. Okay, so here we see, if you read this Marbach et al. Paper, you should see which methods were built on Bayesian networks for matching gene expressions with the stimuli that were leading to them. So you can see that methods 1 through 6, here's one of them over here, are all Bayesian methods that were used in the context of discerning the relationship between expression and stimulus. Okay, move to the next one. In silico docking, oh my goodness, you guys are asking tough questions. All right, in silico docking. So, right. There are a great deal of, there are many software toolkits out there designed to work with data from protein structures. I'm going to start us out on RCSB if I can here. Share screen, show that. Okay, this is the protein data bank using their server over at RCSB. Protein structures are available for a huge number of processes. They have, one of the features I really love here is their molecule of the month, which helps people to understand the molecular mechanisms by which protein complexes carry out different processes. They've also put together a really great resource on how the different proteins of COVID-19 interact. Oh, I heard a beep. What's that mean? Not sure what that beep was, but okay, we'll go with it. So what we see is that a lot of these great resources are available to us. And we also see that, that we have entire toolkits built around the manipulation of 3D structures of proteins. One of the big ones out there is called PyMol, P-Y-M-O-L. It's an entire language, really, for interacting with structures to evaluate what kinds of hydrogen bonds are possible between different binding interfaces. Again, I'm not an expert in it, but my colleague Ruben Klitte is quite good with them. We're actually working on a project together in celiac disease right now because of my interest in that space. Okay, so I don't have a huge amount of information about this, but again, I would say I have recorded a lecture on protein structure determination that's available from my blog post and also just from YouTube. If you search for David Tabb and protein structure, you should be able to find that. So I think that would be the way to go.
So Dominic is asking some questions.
I'm sorry?
Dominic, open பண்ணி விடுப்பா. Thirumular.
The open?
Here. There is a participant is want to ask some questions.
Oh, okay. What, you want to ask them live?
Yeah. Dominic, hello.
Hi, David. I was wondering, now there are different species of viruses and each causing a different disease. Now, if is there any part of the genome that will distinguish one viral species from the other on the surface, apart from the spike proteins?
Yeah, well, yeah, so obviously antigen presentation is one of these primary ways that our bodies come to recognize new antigens. I am not an immunologist, and I don't feel like this is an area of great strength for me.
Okay, a little more specifically, a part of the genome that is conserved, a conserved region of this thing, conserved domains. Will any of the conserved domains also code for envelope proteins?
Will the conserved domains code for what?
Envelope proteins.
Oh,
virus.
Okay. Yes, if you look at the proteins that appear in the outer membrane of these viruses, you'll typically find that they have some regions that are quite static, that may be necessary to forming that particular structure, and others that are quite, that are very poorly conserved. So in a case like that, you would have some regions of the protein that you can rely on for, to always be there, and others that you can't rely on at all.
Yes.
So, yeah, it's possible that we find these very highly variable regions and highly conserved regions all in one protein.
So, when you're looking at a highly conserved region of the proteins, how do you distinguish which? And now we're looking at proteins that may or may not be structurally identified in the genome. So if you're looking at a fragment of the RNA that is coding that you find is consistent throughout all the strains of the same virus, does it I mean, how do you figure out whether it is for the surface of the envelope or whether it is a spike, or how do you determine what exactly that specific portion of the mRNA is coding for?
Okay, I would say that to have a bit of the virus that is accessible to an antibody,
yes,
it really needs to be on the outside of the virus.
Yes, true.
Generally speaking, just because the RNA and proteins are being constructed within cells does not make those cells good targets for the immune system.
No, not the cells, not the cells. I'm talking about now. See, the mRNA has produced the various protein molecules, and
right. So, as I understand it, the coronavirus doesn't have any RNA exposed on the virion.
Yes.
That's all inside the membrane.
Yeah.
So I don't, even if we had a hunter killer that could go destroy any blob that had the viral RNA on it, it wouldn't be of any service because those are inside the virus.
My target is not the RNA, but the fact that every virus, every viral particle that is released from a cell once it is assembled has the spike proteins. It has various other components on the envelope including the mRNA, which is inside, the RNA, which is inside. Now, on the surface, apart from the spike proteins, are there any other components which can be produced by a conserved domain?
To my knowledge, there's only one protein coded for by the viral genome that appears on the outside of the coronavirus. Maybe two? Is it one or two? I'm thinking of flu. Flu has two, the neuraminidase and the hemagglutinin. I don't know for coronavirus anything other than spike protein, but I'm not sure on that one. There may be one or two different polypeptides visible on the outside.
All right. Thank you.
I wish it had a much better handle for us to grab those guys out. That would be helpful.
Because I was looking at the spike proteins, but the problem with the spike proteins is that they're constantly mutating. So I'm trying to figure out any other projections on the surface of the virus which can trigger antibody responses, and whether a vaccine can be created based on this.
There was a really useful article from the New York Times, of all places, that did a quick walkthrough of every protein in the virus. I'm trying to dig this up here. Sorry, it'll take me just a moment to find it. Not that one. It's on this post. Okay, here we go. Click on this. I'm going to share this screen. I really love this article. We don't think of the New York Times as a place that gives us good molecular information, but they published this article on April 3rd that was a walk through all the different parts of it. You can see that it gives an index of all the proteins, ORF1AB, NSP1, NSP2, NSP3, all these different molecules. So this is completely unlike what I expect of the New York Times, but I just thought it was really impressive that they gave this kind of detailed information. So my answer would be to take a quick walkthrough on this. We're clearly learning more and more about this virus every day, so the information we have at hand will only grow.
Can you please share the link with me, or any bit where I can access this site?
Yes.
Because it's really tiny and I can't see it over my phone. New York Times.
Yes.
New York Times and coronavirus.
Yes. You can see the URL there at the top. I guess you can't copy and paste that there. I could put this in the chat session.
Yes, that would be helpful.
All right. I'm going to stop. Well, I'm going to stop this share and come over here.
You see the participant. You click participant.
Okay. You are Dominique?
Yes.
It's not letting me write to individuals.
You send it in chat box.
You can share it on the general chat.
Box. That's these box.
Okay. Let me see if I can get this working. It's now working. Good. Good.
Thank you so much.
Okay. So I've shared two different URLs there.
Yes.
The first one, from picking up the tab, is the list of all the lectures. I've made reference to the microbiome amplicon sequencing lecture and the protein structure lecture. You can find that on the picking up the tab page. And then the second one is the New York Times article that is the walk through the COVID-19 proteins.
All right. Thank you so much.
Sure. Thank you. Let me see. In silico docking, how can we solve the pandemic disease? I think we've kind of covered that already.
It's about COVID-19.
Which protein database would I prefer to blast against for identifying a protein in a new species of bacteriophage? Well, I would note that the NCBI, the National Center for Biotechnology Information, has created a new page for viruses. So if I type NCBI virus, okay, I'm going to give you the URL here. All right. So I've given a new URL to just the virus page. We see that they have an NCBI BLAST tool for finding similar viral nucleotide and protein sequences. So if I simply click into that, enter the sequence, and click Start Search. Oh, am I sharing that window? I'm not even sharing that window. I'm so sorry. Okay, let's try that again. All right. So if I go to the NCBI virus page that I provided the link for in chat just a moment ago, I see that they have accessible a search by sequence tool. Clicking on that, I see that I can enter a nucleotide sequence or a protein sequence, and it will search within the viral collection at NCBI. And yes, it does include bacteriophages. So it's the same tool, it's still BLAST, but instead of searching against genomic DNA of cells, it's going against the genomic DNA of viruses. Okay, I think we're actually caught up on the text questions.
Yeah, you share your email address. You share your email address. That will be helpful for participants.
Oh, sure. My email is dtab@sunacza. There we go.
Thank you so much.
That's where you can reach me. I am often slow to answer my email. I'm sorry about that. Once I shared this index to my lectures, I suddenly discovered that I was mentoring a few hundred people who were shooting me messages via YouTube or via WordPress or via email, and it's been encouraging. I'm glad so many people want to talk, but I'm just one person, so I'll try to keep up.
Nice informative talk.
Thank you.
I would like to call Dr. Tirumula to give a vote of thanks.
Good morning, everyone. I'm happy to share my greetings to Dr. Tab. Very nice, excellent, and informative presentation. Well explanation, sir. On behalf of Annai College of Arts and Science, I take immense pleasure to say thank you to Dr. Tab for sharing your valuable and informative knowledge. Thank you, sir. I thank my friend, convener Dr. Mani, and our principal, Dr. S.P. Monikavasi, for arranging this kind of seminar. Very thank you, both of you. Last but not least, I thank our participants for their patience and nice interactions to the Honorable Chief Guest, Dr. Tab. Definitely, I hope this seminar is very useful. Thank you.
Thank you.
Thank you so much, my dear friend. Thank you so much.
Great to see you again.
Thank you.
Thank you.
Nice to meet you.